Good afternoon, students. I'm back. So today we are going to study the topics, that is development of the male gametophyte and development of the female gametophyte. Yesterday we have generally studied the topic called as pollen grain and also structure of an anther. So pollen grain is generally best defined as a partially developed male gametophyte. It means that for the development of the male gametophyte, the first initial stage is nothing but pollen grain. Right, so let's have a talk about development of the male gametophyte. Right, the development of the male gametophyte generally takes place from a single pollen grain or a microspore cell. Right, so pollen grain is the first initial cell which generally plays an important role for the formation of the male gametophyte. The development of the male gametophyte is endosporic and monosporic. Right? When I talk about endosporic means it generally takes place inside the spore. Right? And monosporic it means that it generally takes place from a single spore. Right? Okay. So now let's divide this topic into the two parts, that is the development of the male gametophyte initiates into two important steps. Number one, that is before pollination in the pollen sac. Number two, that is nothing but after pollination on the stigma. Right, so before pollination, the protoplasm, you can see the protoplasm or the protoplast of the pollen grain undergoes mitosis cell division to generally form two unequal cells. Right, those two unequal cells are generally called generative cell and the tube cell nucleus. The smaller cell is generally called as the generative cell and the larger cell is generally called as the tube cell. Right, generative cell generally contains a large nucleus, right, thin cytoplasm, but has no food and vacuole. When you talk about the tube cell generally has a large nucleus, thick cytoplasm, and contains the reserved food and vacuole. Now, initially when you talk about the pollen grain, isn't it, at the maturity, the pollen grain is generally two cell state structure during the dehiscence of anther. So we all know what is dehiscence. Dehiscence is a process where the anther generally ruptures to release the pollen grain, right? So during that dehiscence of the anther, the pollen grain is usually two cell stage structure. Right, now the after pollination. Now what will happen is that the pollen grain will get deposited on the stigma of the flower and start absorbing a sugary compound called as pectocellulose. So we know that the stigma is sticky in nature because it generally contains a sugary compound. The name of the sugary compound is pectocellulose. Right, now what happens when the pollen grain gets deposited on the stigma of the flower? The volume of the cytoplasm increases in the pollen grain. You can see this is the cytoplasm. So the volume of the cytoplasm generally increases where it generally increases in the pollen grain, due to which it creates a pressure on the intine, right? As the pressure is created on the intine generally ruptures from the germpore and leads to the formation of the pollen tube. Right, so what happens is that the germpore generally gets ruptured and there is a formation of the pollen tube. Now the generative cell, which is the smaller cell, undergoes mitosis cell division to lead to formation of two male gametes. It leads to the formation of the two male gametes. Also the tube cell nucleus enters into the pollen tube along with the cytoplasm. So I can now say that the structure or the pollen tube containing two male gametes, tube cell nucleus along with the cytoplasm is generally called as male gametophyte. So the male gametophyte formation takes place with the help of a pollen grain where the pollen tube is going to contain two male gametes, tube cell nucleus along with cytoplasm is generally called as male gametophyte. Let's have a talk about the second, that is generally called as female gametophyte. Now you know very well female gametophyte development generally takes place with the help of the ovule. Right? Ovule is an egg containing apparatus. It means the ovule is generally going to contain the egg. Right, okay, so in a young ovule, when you generally talk about a young ovule, right, in a young ovule the hypodermal cells which are generally present below, right, they generally differentiates to form the archisporial cell, and one of the archisporial cell during the maturity leads to the formation of megaspore mother cell which is diploid in nature. Right, it leads to the formation of a megaspore mother cell which is diploid in nature. Now this megaspore mother cell undergoes meiosis cell division to form tetrad megaspore. right? This is generally called tetrad megaspore. You can see 1, 2, 3 and 4, right? Now out of this 4 tetrad megaspore, right, the lower one is generally called as the functional megaspore, correct? And the upper 3 are generally called as the non-functional megaspore, right? So the lower one, functional megaspore, the upper 3 non-functional megaspore. Now this functional megaspore will undergo growth. It will enlarge in size. It will undergo growth and it will enlarge in size, and further will undergo mitosis, cell division. So the functional megaspore will enlarge in size, it will gain the nourishment, right? It will grow and it will undergo repeatedly mitosis, cell division to form your 2 daughter nuclei, 4 daughter nuclei and 8 daughter nuclei respectively. Right? So functional megaspore undergoes repetitive mitosis, cell division. It leads to the formation of 2 daughter nuclei, 4 daughter nuclei and total 8 daughter nuclei present in a embryo sac. Now this upper 3 non-functional megaspore undergoes degeneration, and that's why they are generally called as degenerative megaspore, right? Now you can see that a embryo sac is generally formed having total 8 daughter cell. Having total how many daughter nuclei or the daughter cell? That is nothing but 8, right? Okay, that is 4 present in the upper pole and 4 daughter nuclei present at the lower pole. Now one of the daughter nuclei from the upper pole and one of the daughter nuclei from the lower pole will come at the center, will fuse with each other to form secondary nucleus, right? So this daughter nuclei from the upper pole, this daughter nuclei from the lower pole, they will come in the center, they will fuse with each other and they will lead to the formation of a secondary nucleus which is diploid in nature. That is, double the number of chromosomes are present in the secondary nucleus. Now, how many daughter nuclei are present at the upper pole? 3. How many daughter nuclei are present at the lower pole? 3. So the daughter nuclei present at the chalazal end will lead to the formation of antipodal cell, right? Okay, and the 3 daughter nuclei which are generally present at the lower micropylar end will lead to the formation of egg apparatus, right? So 3 daughter nuclei present at the chalazal end will lead to the formation of antipodal cell, and 3 daughter nuclei present at the lower micropylar end leads to the formation of egg apparatus. Now, these antipodal cells are generally degenerative. Once the fertilization takes place, they undergo, that is, they generally undergo degeneration, and that's why they are generally called as degenerative cell, right? Okay, secondary nucleus is diploid in nature, right? Okay, and the third one is nothing but the egg apparatus. Now, inside the egg apparatus, the central cell is generally called as the egg cell, and the 2 flanked cells are generally called as the synergid. So the central cell is called as the egg cell and the 2 flanked cells are generally called as the synergid cell. So now the structure containing 3 components, that is, first antipodal cell, second secondary nucleus and third egg apparatus along with the cytoplasm together is called as embryo sac or the female gametophyte. So the structure containing 2 male gametes, that is correct, not 2 male gametes, the structure generally containing antipodal cells, secondary nucleus, egg apparatus together is generally called as embryo sac or the female gametophyte, correct? Now this embryo sac or the female gametophyte is 7-celled 8-nucleated structure, correct? That is, total 7 cells are present, 1 2 3 4 5 6, correct now, and 7. So 7 cells are present and total 8 nucleus are present, 1 2 3 4 5 6 7 8. So it is a 7-celled 8-nucleated structure. Such kind of embryo sac is generally called polygonal type of embryo sac. Thank you.